One is neuromuscular control. The axons of the nerve cells of the spinal cord branch and attach to each muscle fiber, forming a neuromuscular junction. An action potential passes down the nerve. The nerve then releases calcium that results in the release of acetylcholine. Step 2. Acetylcholine is released and binds to acetylcholine receptors in the sarcolemma. For step 3, acetylcholine binds with receptors and opens sodium channels. Sodium channels open and sodium comes in, and there is a decrease in the resting potential. Sodium rushes in and the sarcolemma depolarizes, and the regional depolarization spreads rapidly. Potassium channels open and the region repolarizes. Immediately after the action potential passes the membrane, permeability changes again. Sodium channels close and potassium channels open. Potassium rushes out of the cell and the cell repolarizes. And then for step 4, calcium is released from the sarcoplasmic reticulum. Calcium is stored in the sarcoplasmic reticulum. Depolarization releases the calcium, and then the calcium clears the actin binding sites. So step 5 is sliding filament theory of contraction. The first part, the myosin head attaches to actin, and for the second part, it's the power stroke. The myosin head pivots, pulling the actin filament towards the center. Next, the cross bridge detaches when a new ATP binds with the myosin. Cocking of the myosin head occurs when ATP, ADP plus P occurs, and another cross bridge can form. And the end result is the shortening of the sarcomere. The distance between the Z discs shorten, the H zone disappears, the dark A band increases because the actin and myosin overlap more, and the light I band shortens. Calcium is removed from the cytoplasm, and tropomyosin blocks the actin site.